We had completed our sections for the NDA to file on the CMC section, have a rolling NDA, and we were planning on filing in 2008. This was all done over 8 years, and I went from 5 milligrams to 35 kilograms a production run. The cost of goods fell from about $138 to about $20 a vial. So it gives you a sense of how much the drug actually costs. It's not very much in the big scheme of things, even though this is a very expensive drug. And finally, this is such a unique patent that I got a patent on composition of matter patent on the final two intermediates in the synthesis as well as the synthesis. Now you can say, well, how good is that patent compared to the composition of matter you actually have on the drug? And it's part of what we call a picket fence strategy. It just makes it more difficult for people to come in when the drug goes generic and manufacture it. They can't use exactly your method, and that has all kinds of ramifications in terms of protecting the product. Now we created a formulation, and this is the formulation flow, and it's essentially sodium citrate citric acid buffer. Telcyta is a very acidic molecule. It's about pH 3 in solution, and it oils out in solution at pH 3. That's an intolerably low pH to inject. And so we needed something to at least bring it up closer to neutral. In addition, it's not soluble. So if you're going to have it in a formulated vial, you can't have it at just the drug itself. You need to kind of buffer it so it goes into solution. On the other hand, as I told you, at pH 5, Telcyta begins to fall apart. And so we had to set it up in such a way that we started at a higher pH buffer, and as we added the drug, and we had to add it fast, and we had to whip it around with a high-speed mixer so it would dissolve and bring the pH down from, I think it was 6.5 down to 4.5, all without warming up, which accelerated the decomposition, all without decomposing the drug. So by the time we were done, we had a nice drug solution that we could then, we had to show that was sterile. Now finding manufacturers that will do lyophilization of cytotoxics and give you sterile product is a very tricky business. The FDA pays actually much more attention to these people than they pay to the API manufacturers. Because if you have a contaminated vial, you can have all kinds of problems because this is an injectable drug. Also, cytotoxics tend to have safety issues. You have to be really careful around cytotoxics, and a lot of companies just won't handle them. In the end, because this was a unique process and because of the particulars of the molecule, I actually got a granted patent on a citrate buffer formulation. So when we talk about patents on formulations of yesterday's drugs for tomorrow, it's doable, but it has to be special. And again, this is such a special process that it does afford us a certain amount of protection against the generics. So here's the product, here's a picture of the vial itself. And it was meant to deliver about 1/4 gram in, I think it was 5 mils of solution. We'd take those, figure out what the dose was for the patient, withdraw this material, put it in a bag, and it was infused. The cost ultimately for a 40,000 vial run was about $50,000. So not very expensive in the end for the manufacturing. Because we were going to go into production, and our contractors didn't really have dedicated production equipment for us, we decided to manufacture a dedicated kettle. And for a mere $80,000, we got this built. And again, we did all this before we knew the drug would actually succeed. Specifications. So a lot of what the FDA is interested in is, did you make what you said you made? And is it consistent from batch to batch? So we have a quality attribute, which is any number of characterization attributes. We have acceptance criteria, and these reports, this is an older C of A, these get filled in and defined. As the process matures, it becomes tighter and tighter specifications, which means there's a greater and greater chance of failure. And, gee, we missed a slide somewhere. There was $1.5 million production run. Somehow that slide disappeared. I brushed through it. And so since I was putting into play $1.5 million, you don't want the batch to fail. It's somewhat recoverable at the API. At the stage of the drug product itself or the formulation, it's no longer recoverable, and you dump it. So making sure we met these criteria as they became stricter and stricter, and that's based on statistical analysis of the number of batches. And so you're caught between wanting to make a lot of batches to get real good sense of how much control you have over your product production, and the CFO saying, no, you can't produce these batches. And because you have no use for them, you're going to throw them away. They're not part of our commercial launch. And so this becomes a guessing game a little bit as to how tight you want to make that. And it was a constant argument between myself and the quality people as to how tight we should make these specifications. In addition, we spent about, of that $30 million, we spent about $4 million on just analytical method development. And we spent $2 million on stability studies. So you get a sense of what these kinds of development costs run. So here is our supply chain. This is the producers of the TPE alcohol and the tripeptide. These were going to be the qualified producers and test labs for the API. These were the manufacturers of the drug product or the formulated product. These were the test laboratories. This was the distribution. This was a distribution center. And this was an old distribution center that we didn't use anymore. I did show it. So this is what a $1.4 million production looks like. It's those bags, Mylar bags. Forgot I took it and merged the slides.